Yeah, should I wear the left light? True. Okay. All right. Okay. All right, welcome back, everybody. This is more people than I expected for this lecture, but glad to see you all here. Okay, so today's learning objectives. So we're going to talk about the basics, and you probably have covered some of this in your PBL stuff, but we're going to talk about the molecular stuff, my favorites. So we're going to talk about receptors, ligands, and targeting, and then we're also going to talk about membrane receptor signaling. Like there's many different types of hormone receptors, but today we're going to focus on membrane receptors. And then, and also not just membrane receptors, we'll talk about one of my favorite subjects, nuclear receptors, which I did my master's and my doctoral studies. Like I was trying to target estrogen receptors, none of it worked out. But I learned a lot about it along the way. So sometimes it's about the journey, not the destination. All right. So you know my legend that I borrowed from or adapted from Dr. Matsui. So with that, and we covered about the hormone classes. So I know different textbooks have different ways of determining it, but the way I group them are the monoamines. So again, made from one amino acid, and the only ones we know about are the ones made from tyrosine, like all the catecholamines, or tryptophan. And then we have our peptides/proteins. What's the difference? Well, one's long, one's short. And then we have our lipid-soluble ones, which are mostly steroids. So monoamines, since they are amino acids, small molecules, they circulate freely. So they're water-soluble. And same with the peptides and proteins. They are generally water-soluble, and it makes sense because they have to be carried in the bloodstream. And because and the cool thing is this is also why they can be stored in vesicles as well. So with that, the thing about that is since they're water-soluble and hydrophilic, they have a hard time crossing barriers, and especially that phospholipid bilayer that our cells are surrounded with. Now then, steroid and thyroid hormones. So again, thyroid hormones, and this is why I always emphasize to the undergrads monoamines, because technically thyroid hormones are made from amino acids plus iodine. But the thing is that once you have thyroid hormone fully assembled, they start behaving more like steroids, and I'll explain why later in this lecture. All right, so here we have a bunch of steroid and thyroid hormones. So the thing is that steroids, well, things that are oily and things that are hydrophobic tend to clump together. So what prevents them from clumping together with other types of lipids and hydrophobic molecules? Well, they're pore soluble, and plus, would they float freely in our blood, which a big component of it is water. So then you have globulins and albumins. So you have all these proteins that are hydrophilic. And the cool thing is that, especially for things like sex hormone binding globulin or thyroxine binding globulin, they have a little hydrophobic pocket that these steroids or thyroid hormones can bind to and hitch a ride on. So even though you have this hydrophobic hormone and a little hydrophobic site on a globulin, the overall thing is still hydrophilic, this whole overall complex. So this is how these steroid and thyroid hormones can circulate and prevent themselves from being lost. It's kind of like an emulsifier for these steroid and thyroid hormones. But even albumin is kind of interesting. It's kind of like a sticky protein because many hydrophobic, it's very non-specific. Things like fatty acids can stick to albumins, or even ions can stick to albumins. And same with these steroid and thyroid hormones. These lipid-soluble hormones can bind to albumins. So then, so we have sex hormone binding globulins, such as the one that transport, and they pretty much transport these categories of hormones pretty evenly. thyroxine binding globulin, so binding the T3 and T4, transcortin for glucocorticoids, and the thing about that is that these need a way of getting through your blood and your bloodstream and being transported throughout your body. So they need to, like the vast majority of our steroid and thyroid hormones, they are bound to these proteins. Now the thing is that this overall hydrophobic hormone plus protein, transport protein complex is hydrophilic. Because it's hydrophilic, then the steroid can't actually get past the membrane. So when they're bound to these transport hormones, they are not available. So what happens is like biochemistry needs to, and some of these dissociate, so it needs to dissociate from the transport hormone to actually be released and freeze so that it can diffuse into whatever target cell has the receptors for these steroids and thyroid hormones. So the other thing is that this is another feedback mechanism because say the body's like, hey, I'm getting too much of this androgen signaling. What's one way it can do? Well, it can shut off the HPG axis to prevent further synthesis or prevent further stimulation of androgen production from whatever reproductive organs. But it can also secrete more of the sex hormone binding globulin to kind of like absorb and kind of keep these androgens in check. So it's kind of like another feedback mechanism in addition to your hypothalamic pituitary feedback. All right, so then hormone, why do you make a big deal about the solubility of these hormones? Well, it also affects the drugs that target them and the receptor location. So since our monoamines and proteins are water soluble, well, they can't cross the barrier of our plasma membrane. So it makes sense that their receptors would be on the membrane facing the extracellular space and interstitial fluid, where you find these monoamine and protein hormones floating around. Now, cytoplasmic receptors and nuclear receptors, I see none yet because that's the thing. It's like it's always they find a new pathway, they find a new molecule, but until they find one way of like these water-soluble hormones actually transporting into the cell, which I haven't heard of it yet, they don't have these receptors. So when they bind to these monoamine receptors or protein or peptide receptors on the membrane, this causes intracellular signaling, and that causes the release of molecules or the production of second messengers. So, and this is a funny thing, like throughout my entire undergrad, like they covered all things like GPCR, membrane receptors. They said second messengers. So I was like, what's the first messenger? So the first messenger is actually the hormone itself, and the second messenger is after the receptor. So I know I only learned that after undergrad, but I started teaching that. Okay, so then steroids and thyroid hormones. So the thing is that here we have a steroid, here we have a thyroid hormone T4. So they have cytoplasmic receptors and for steroids or certain steroids, I should say. So here we have a steroid with a cytoplasmic receptor, and what typically happens is that when it's inactive and there's no steroid, these receptors are in cytoplasm. But once they bind to their target steroid, then the whole complex with the steroid and its receptor moves from the cytoplasm to the nucleus, where it binds to promoters and activates genes. And then nuclear receptors, well, there are some nuclear receptors that actually they just stay in the nucleus. So actually rewind a bit. So here's an example of gene regulation. Now, thyroid hormone receptors, the classic genomic actions where it activates genes. The thyroid hormone receptor alpha stays in the nucleus, and then thyroid hormone is transported into the cell and transported from the cytoplasm to the nucleus. And it can't do that because thyroid hormone is lipid soluble. And then it's going to bind to promoters and activate genes. Now, the thing is that, and it's not really new knowledge, but there's also membrane receptors for steroids and thyroid hormones. So they do have membrane receptors, but it's going to also cause second messenger release. So the interesting thing about steroids and thyroid hormone is that they kind of are able to do these genomic actions, but in general, second messenger mechanisms are faster than having to bind to DNA, activate promoters, recruit other transcription factors, recruit co-activators, recruit all the machinery to cause RNA transcription. that is pretty slow, but these are pretty fast mechanisms. So a lot of the steroids have a slow genomic mechanism and a fast second messenger mechanism, and same with thyroid hormone. Why don't you typically learn about this? Because this is my prediction. Once they have a drug that targets a steroid membrane receptor or a thyroid membrane receptor, then you probably have to learn it. But right now we don't have any. So the thing is that, so I like to use this with receptors and ligands, why I make a big deal. Well, things that hormones bind to receptors, and that causes some change in the receptor and their shape. So I like to think of it just like a lock and key and ignition. So we put the key, and what does it do to the pins? It changes the shape of things in the pin, in the lock tumbler, and that's what we see with this key in the lock. Turn it, and then you get some sort of change. And what does it do? Well, if this is an ignition to the car, it's going to start the car up. Now, with hormone targeting, the thing is that what if that key doesn't fit? You can't do this. So you can't turn, you can't have action. So with hormone targeting, you need a fit of this hormone to its receptor. So here's the little things, like here, we have different types of hormones. If it doesn't bind to the receptor and fit into its pocket, it's not going to activate the receptor. So the thing is that our blood distributes hormone throughout the body, and blood goes everywhere in our body, right? But not all cells respond to every hormone, despite all the hormones that are circulating in our blood right now. So how do you control it? Well, your body is able to target certain cells with certain hormones by having certain cells only express receptors for hormones. So it's kind of selective that way. So if you want a hormone that has a wide variety of effects and a very powerful effect in the body, like thyroid hormone, you're going to find thyroid hormone receptors throughout the entire body. But if it's going to be kind of specific toward an organ or tissue, like say ACTH and the adrenal glands, well, you want to keep those ACTH receptors within that adrenal gland. Or who knows, then they'll find some ACTH receptor somewhere else, and then I have to write my whole lectures over again, but luckily we haven't found that. The thing is, like, they always say, like, oh, we found another pathway, which is cool, but it makes things like, yeah. So, all right, so the thing is that it's not always one-to-one. So it's not always like just one hormone and only one receptor binds to. There is, like, it goes both ways. Sometimes you have multiple hormones binding to the same receptor, or you could have the same hormone binding to multiple receptors. So here's an example like estradiol. So estradiol is the main estrogen. So typically if somebody's saying like estrogen singular, they're probably referring to estradiol. Now estradiol, so the classic one, if they talk about classic estrogen receptor signaling, you have the estrogen receptor alpha, which is a cytosolic/nuclear receptor. But there's also an estrogen receptor beta, which is kind of like I would say maybe the sister that behaves, and it's kind of interesting. They thought it was a repressor. They said, no, she activates in other tissues, and then, yeah, ER beta is kind of interesting. And then they found a membrane receptor. So this one's actually pretty hot in terms of like research, and so there's a G protein coupled receptor 30s, or this is a G protein estrogen receptor. All of these different proteins, but they bind to the same ligand, and they're all activated by estradiol. Now, the thing is, like, what about the opposite? So here, interesting is that we have estrogen, in this main estrogen, but multiple receptors. But for androgens, there's only one known nuclear receptor, androgen receptor. They have found a membrane receptor for androgen receptor, but it's less known. So here we have like, 6 different flavors of androgens, but they all bind to the androgen receptor. Now, they're not all equal, and I put these in roughly kind of, the bigger the arrow, the thicker, the more potent of an activator is of the receptor. So some of these activate the androgen receptor, but things like DHT, dihydrotestosterone, is like super testosterone. It's it's a very potent activator of the receptor. But either way, same receptor, just different, but many different ligands can activate it. And then, let's see, so then we have pharmacophore. Now this is an interesting, I don't really see this in medical textbooks, but it's borrowed something from pharmacology and toxicology. So pharmacophore is like the minimal features you need for so it's just what chemical groups do you need to actually tickle the receptor and activate it. So like was it? So I like to think of it like a lockpick, because the thing about lockpicks and the typical tumbler and pin type of lock right here, sometimes you only need to tickle one of these pins. And just like this main one, if you just do that, you're able to move the whole thing. So I like to think of the pharmacophore like that. It's minimal, it's not the full key, it's all you need to actually cause that change. So, or like, what was it? I know it was like, maybe, has it been that long? But there used to be this show called Orange is the New Black. And then there, okay, so you have seen it. But you know, like those two inmates, and then they take that security guard and they have to get like the fingerprint through, and they want to get his fingerprint to go through a door. So what they do, they, instead of getting the whole guard, they take part of the guard, right? And they use that to open the door. So I think of pharmacophore like that. You don't have the whole thing, you just have all you need to actually unlock the lock and get to where you need to be. So why are we going to talk about pharmacophores? Well, we have estradiol and our estrogen receptor, but there are other things that act like estradiol. So here we have tamoxifen from MD1. And then tamoxifen is like, it has these forms, like especially the ring structures. That's kind of this pharmacophore it needs to fit into estrogen receptor alpha. And then we have lacustrant. So here we have like similar features right here. Like here, estradiol, we have these hydroxyl groups. Not quite hydroxyl here, but we have rings and a hydroxyl group that kind of acts like it for the pharmacophore. And then here we have licoritiginin. So this is actually cool because it's like a plant compound. And licorice. So licorice, this is why like licorice has, and sometimes you may see in one of those zebra questions, like they're not eating licorice because licorice actually activates so many receptors. I mean, it's not like as powerful as estradiol, but it does have kind of similar shape, and it does have these two hydroxyl groups. And then bisphenol A and BPA. So if you've had a water bottle, you might hear like BPA free, because why? Bisphenol A is a plasticizer. So it's found in plastics, and this is why a lot of these plasticizers are kind of under investigation. Well, who knows, but these days. But either way, simple compound, but you notice that it's kind of like able to fit into the binding active site of estrogen receptor thanks to these minimal features. All right, so this little disclaimer here. So when I teach these pathways, I teach them kind of like this so that everyone doesn't go insane. But it's like complex, but it goes from point A to point B, right? So, but the thing about endocrinology and why I always have to read it and update my slides every year is that in terms of like signaling and these pathways, it's actually more like this. Where they all cross-talk with each other. So you think about hormones and these enzymes, they do not stay in their own lane like that previous image. It is something more like this. So that's why I keep it simple and teach it like this. Because otherwise that's my main point. Cell signaling pathways are complex, but I'll walk you through them, so the ones we covered today. All right, so one of the main ones we'll cover today are G protein coupled receptors. And this is like a huge family. Like there's so many numbered ones, and I don't know why they have me memorize some of these ones, but it's just a huge family. But what they have, thankfully, they have things in common. So you might remember all the way back from like maybe your sophomore biology or MCAT study. Pass through the cell membrane 7 times over here, and they form this kind of structure like this. All right, yep. So G proteins, what they do, what's the G? Well, this whole thing right here is a G protein, and then it has subunits, the alpha, beta, and gamma subunit. But G proteins, they bind either GTP or GDP. And then the things that the G alpha subunit binds GDP or GTP. So when it's bound to GDP, it's inactive, and when it binds to GTP, it's active. So the way I remember it is like ATP, that has energy, ADP doesn't have energy. Well, it's just change A for G. So in terms of this, we have the energy with the GTP, active, GDP, inactive. So it's kind of like the on-off switch of the G proteins. The thing about that is like, again, my molecular biology, they're always discovering new things. There's multiple subtypes, so it's not just like about G protein, the alpha subunit, you also have subtypes of the different types of alpha subunits. But the thing is that these subtypes, they act differently. So you might have the same G protein coupled receptor or different ones and G proteins, but they do different things based on what type of subunit you have here. All right, so example are our adrenergic receptors. So here we have our catecholamines such as epi, norepi, and dopamine. And then we have our alpha 1, alpha 2, beta 1, beta 2, beta 3. Well, so things that they use second messengers to carry out their effects. And here we have adenylyl cyclase. So here we have the G protein. Activate these G proteins, and they activate the GPCR. They activate the G proteins that activate this enzyme that triggers the production of the second messenger, in this case, cyclic AMP. Now, the responses are based on what receptors? So different receptors, different effects. So like alpha 1, vasoconstriction, alpha 2, among many things, it will decrease GI motility. Beta 1, so that one, classic one where it increases heart rate and contractility. Beta 2 is in the heart as well, but beta 1 is the main one, the dominant one. And beta 2, smooth muscle relaxation, that leads to bronchodilation. But I remember like one year one student said like, wait, dopamine is stimulatory, and in this case, how is it both stimulatory and inhibitory? Well, the receptors are different. So you have the same molecule, but say you want it to be stimulatory. You are not going to have this dopamine receptor 2 on the cell, so that dopamine is just stimulatory. But what if you want dopamine to be inhibitory in the cell? Well, it's not going to have this one here. It's going to have only this receptor. So this is how the cells are able to. So you might have the same receptor, you might have the same ligand, but it might have different effects in different cells because these cells have different receptors with different effects. So classic example is like tamoxifen. Like tamoxifen is an antagonist in breast tissue, but it's an agonist in things like endometrial and uterine and ovarian tissue. So it depends on what receptor is and also what second messengers and mechanisms you have inside that cell. All right, so just like a little review of GPCR signaling, you may have studied for the MCAT. So hormone or ligand binds to GPCR, so it could be a drug that acts like a hormone. And then this causes changes in shape, just like the lock and tumbler, and allows G proteins to bind. And then G proteins, when they're activated, they are going to swap GDP for GTP. And when it's activated like that, then this alpha subunit is kind of like, okay, I got my GTP and it breaks up the group and it's going to go solo and go off do its own thing. Now, one of the things it can do is go to, like for the G alpha S family, so this alpha S subtype, it's going to activate adenylyl cyclase. So adenylyl cyclase produces cyclic AMP, and then from ATP. And then you have the GQ family. The GQ family is going to produce or activate phospholipase C. So we still have GPCRs, but different types of G alpha subunits. And then phospholipase C is going to produce IP3 and diacylglycerol. So there are other types, and this one is just like, like the GI family, and I think they used to call this like G monk or something, and they probably found many other ones, but thankfully, or actually, maybe of these, the GI family is the most studied of that one. So usually it's inhibitory, but of course, like molecular biology, they always find some alternate pathways. And there's other less studied ones, but GS and GQ, they're pretty well known. All right. So speaking of GS. So GS, alpha S subunit, is going to activate adenylyl cyclase. And adenylyl cyclase is going to take ATP, get rid of 2 of the phosphates, and make the whole thing a circular cyclic structure. You have cyclic AMP. And then the cyclic AMP is the second messenger. And the second messenger, in this case, the classic cyclic AMP effect, is going to bind to inactive protein kinase A. And protein kinase A is interesting that it has multiple subunits. So it has a regulatory subunits and a catalytic subunits. So when a cyclic AMP binds to the regulatory subunits of protein kinase A, so right now it's inactive. But when you have cyclic AMP, it's going to bind to the regulatory subunit. And then this is going to trigger changes in shape that allow that catalytic subunit to go free. And what happens next? Well, just depends on the cell type. So this can go on to phosphorylate and activate other proteins. So the way I like to think of it is kind of like These are two like little dogs ready to play, but they're held on a leash. But what happens when you let the clasp go? They can go off into the doggy park and play and do whatever they plan to do. But again, just like different dogs have like what do dogs do? Well, it depends on the dog, depends on the breed, depends where they are. Same with the protein kinase A. So the thing is that protein kinase A, it's going to, yeah, so this kind of summarizes this in this figure. Now, phospholipase C, this is with the G-alpha Q subunit. So the way I like to think of it is like there instead of just cyclic AMP being the main product. So here we have PIP2 and here we have phospholipase C, and notice that this has these fatty acid tails. So PIP2 is stuck in the membrane, and phospholipase C is also attached to the membrane. So the way I like to think about this is like it's kind of like this is like a little head of this PIP2, and these are like the little legs. And phospholipase C is like a little axe or guillotine and just cuts off this little head. So you have the little head that's floating free, but the legs are still stuck in the membrane right here. So diacylglycerol is most of that. It still has that fatty acid tails right here. So it stays in the membrane and can also activate protein kinase C right here. That's usually hanging out near the membrane. So lipid-soluble factor that can diffuse now that it's like free from its inositol triphosphate. And this will activate protein kinase C. So cyclic AMP activated protein kinase A. This is a different flavor of protein kinase, but it's C. And then it's also going to, but that head isn't just like inactive by itself. So this phosphatidylinositol, the PIP2, is just going to, the diacylglycerol is one part, IP3 is the other part. So this inositol triphosphate is water soluble, and because like it's just like it has all this phosphate group, so it has a lot of charge. If there's a molecule that's pretty polar or has a lot of charges, it's generally water soluble. And what this does is bind to its own type of channel and receptor. So this one's kind of cool. It's both a receptor and a channel. And then you find all these IP3 sensitive calcium channels in the endoplasmic reticulum. And the endoplasmic reticulum, you might remember this from MD2, is a big storehouse of calcium. So if your cell wants calcium to activate all calcium signaling inside the cell, you'll release it from the endoplasmic reticulum, or you can store it for later. But in the case of IP3, typically it's going to bind to this and cause calcium release. And it can activate other proteins such as calmodulin. And yeah, or other voltage gated ion channels, yeah, because again, you have like this +2 charge, so you're introducing a whole big change in voltage as well when you release calcium. All right, muscle contraction, yeah, if you have actin myosin, what does it need to bind? Well, troponin needs that calcium. So if you have like a muscle cell and a lot of IP3, it's going to trigger contractions. All right, so to summarize the ones we know about, so G-alpha S subunit is going to activate adenylyl cyclase, which is going to trigger the production of cyclic AMP. Cyclic AMP, the main target of that is protein kinase A, and downstream effect, well, protein kinase A is a kinase, so it'll phosphorylate other targets. Depends on what targets, depends on the cell. So this is my mnemonic, so I like to think of it, so S plus here, and this is why I showed like that protein kinase A kind of scampering off, because so I have to put S plus camp, so like scamp. So it scampers off this protein kinase A for G alpha S. So I just put S and camp together. And then another one, or another mnemonic was like summer camp. So S and camp together, that means like J alpha S and camp, cyclic AMP. Now J alpha Q subunit, so that is going to give you 2 second messengers. You have the one that stays stuck in the membrane, you have diacylglycerol, and then you have the previous phospholipid head, so inositol triphosphate. Diacylglycerol, the primary target is protein kinase C, and that's going to also activate and cause phosphorylation. Inositol triphosphate is going to bind to its own receptors in the endoplasmic reticulum, triggering calcium release, and calcium does many things, including calmodulin, which we don't really have time to talk about today. So this one, several years back, and I keep on mentioning this one because I remember like one of the class of like 2021 is like, you know, you have like the stupidest mnemonic, but I still remember it to this day, and I did encounter it on my step 1. So I'm going to teach you that mnemonic right now. All right. So I had a photo shoot for GQ, but I was so nervous, and it was an early morning photo shoot, so I drank so much coffee that I had to pee 3 times. So GQ, dang, and I pee 3. So that's the way I remember that one. Yeah, he still remembers it. So I'm like, hey, it's stuck in your head. That's the whole purpose of it. So yeah, so it worked for him. All right. So that is like GQ subunit. Now let's talk about so there's other types of receptors. So like the guanylate cyclase, and then that produces cyclic GMP. And I give you examples of like hormones that use these receptors. Serine threonine kinase receptors we don't really cover today. We don't have time. So here are examples of that. Now let's cover receptor tyrosine kinases and example hormones and ligands that target these type of proteins are things like insulin and insulin-like receptors. So insulin receptor signaling, and I think this is still pretty much the same from first aid. Yeah, so luckily this is what you have to know for med school, and even things like GLUT4, like you have to know this isoform of this glucose transporter. But if you do research, it's more like this. So luckily you don't have to memorize all of that. So I'm really glad that you only have to. This is kind of like what the key right here. But yeah, so like this is a basic insulin cellular mechanism to kind of make it simple. So glucose, beta cells in your pancreas, they detect glucose, and you probably learned about depolarization of potassium and other sorts in your studies. But for now, just know that they detect changes in glucose. And when they detect high glucose, that causes changes that cause them to release insulin. Insulin receptors on cells that have them, when it activates that triggers multiple enzymes, but one of the big ones is it's going to cause glucose transporters to move from inside the cells. So it's kind of like storage. When you don't have insulin, they're like in storage. But once you have insulin and activating insulin receptor, this causes them to appear at the surface. And now that I have glucose transporters at the surface, now this glucose can move in. And the thing is that this is before and after. So by moving glucose into a cell from the bloodstream, that lowers the overall blood glucose levels. Yep, so keep on doing that. Yeah, you lower that blood glucose. All right. So then here we have beta cells, but hey, what if we have type 1 diabetes? What's the main mechanism? Well, here we have a bunch of ganglion immune cells, and for some reason they have something against the beta cells of the pancreas. Yeah, really violent immune cells, but then so the poor beta cells, they're all beat up and they can't produce their insulin anymore. So then what happens? Well, if the beta cells can't produce insulin, can we activate the receptors? It's like having your car but no keys, so that there's no insulin. So no insulin means no insulin receptor activation, no appearance of these glucose transporters on the cell surfaces. So glucose pretty much has no way of moving into these cells. So glucose will build up. And that's why with uncontrolled type 1 diabetes, you can have this hyperglycemia. Now, the other thing is that hormone receptors, there are multiple ways of your cells adjusting the signals as well. So here we have hormone and receptors, but say your cells want more of the signaling. So what can they do? Well, what they can do to make a hormone more potent is that if you have more receptors on a cell, then they have more of this enzymes being activated, and more enzymes being activated means more activity. So if your cell, things that your cells, they have specialty and they know to look for these hormones, but they say, I should be getting these hormone signals, but I'm not getting enough hormone signals. So this is one way they can try to get back to homeostasis and get the appropriate amount of hormonal signaling. So they can increase the amount of receptors and their activity. And then this increases sensitivity. So if you want to make a cell more responsive, one way is to increase the amount of receptors out on the surface. But yeah, so that's what can happen after a hormone is deprived in the body. Now, the opposite can also happen as well. It's like, what if the cell's like, okay, I shouldn't have this hormone signal all the time. Let's dial back on that. So it can also remove receptors from the surface and downregulate. So less receptors means that less enzyme activation. So this, even though we didn't really change the hormone level in this case, just by removing and making the number of receptors fewer, then you have less activity. So this will reduce sensitivity. So more receptors on the surface, increase sensitivity, less receptors, less sensitive. Now I bring this up because this is one way that you can have insulin resistance. So what happens if your cells don't have the receptor or they express fewer receptors? Well, fewer receptors, you still have the same amount of insulin in this example, but with less receptors and less cells that are sensitive to these, or have these receptors, you have less cells that are sensitive to insulin. So here we have this poor cell by itself, the only one with the insulin receptor, and will it be able to do the work of 5 previous cells? It's going to have a hard time keeping up. So it's going to still absorb some glucose, but it's going to, you still have, say the next meal comes along. Well, the beta cells are still there in the background, still detecting, hey, there's a lot of glucose here. Let's increase our insulin. So it's going to send insulin, but all this insulin is here, but you don't have enough receptors and cells with receptors to activate that. So what happens is that we have this insulin piling up in addition to this glucose. So that's one key difference between type 1 and type 2 diabetes. Type 1 diabetes, beta cells aren't producing the insulin, but type 2, at least in early type 2 diabetes, you have the beta cells are there. And they're detecting that elevated hyperglycemia, and they're saying, like, okay, we're going to increase our insulin production. They don't know that the cells downstream aren't responding to it. So I have a better version of this. Okay, yeah, here. Okay, so insulin receptors. So things that insulin receptors, they cause many things, but things that, interestingly enough, insulin receptors also inactivate themselves. So they also have their own negative feedback mechanisms. And yeah, so things that if you have chronic insulin receptor activity, it can actually shut it off even though you still have insulin there. And one of the mechanisms is, things that this insulin receptor shuts off its own activity, glucose transporters no longer appear at the surface, and glucose will pile up, and then beta cells will still crank out insulin to have insulin, and so insulin will also pile up as well, leading to hyperinsulinemia. Okay, so desensitization. So here we have insulin, we have receptors, but say these mechanisms of insulin receptors turning themselves off. Well, you have less glucose transporters, and therefore we'll have glucose piling up, and we'll also have insulin piling up as well. So do you need to memorize this pathway? I'll walk you through it. And this is one of the ways that insulin receptor can turn itself off. So I'll guide you through this. So this is one way of activating all these enzymes are here. So you have insulin receptor, IRS1, AKT, but then it activates this protein called mTOR, and mTOR is actually part of a huge protein complex, and I spent like a few years of my life studying. But interesting, like mTOR right here, it's going to activate another enzyme, but this enzyme turns off insulin receptor. So it's not a direct pathway, but eventually, if you have chronic insulin receptor signaling, it's going to activate all these enzymes, including mTOR, but mTOR will turn off insulin receptor eventually. So another way, it's not just insulin receptor turning itself off. This is also why diet's important. Here we have free fatty acids and free fatty acid receptors, multiple types. So free fatty acid receptors, when they bind to, or free fatty acids, so someone has a high diet rich in triglycerides and fatty acids, can activate these receptors, and I'll trace the pathway right here. And then we have protein kinase C epsilon that inhibits the IRS one right here. So that is going to inhibit this substrate and inactivate all this signaling down here as well, and including the signaling that would lead to this glucose transport. So it's not just like insulin receptor turning itself off. You can also have like things like hyperlipidemia turning insulin receptors off. So an analogy I like to use is, say here we have like you're trying to listen to you have a party and you want to have music at your party, and you're trying to stream it off from whatever streaming website. So here we have a transmitter, and it's sending the signals with all your music. So I think that signals kind of like the hormone, and then you have receptors, which are your speakers. But if you want to increase the volume of it and make that signal boost that music, what you're going to do, one thing you might do is add more speakers that receive that signal. So you still have the same signal, but by increasing the amount of things that receive that signal, you also increase the overall activity. But say it's too loud and you want less and less signal right here, what can you do? You can decrease the amount of things that receive that signal. So, but desensitization, I like to think of it this way, it's kind of like, what if these are broken? So if someone cuts the wires, that's one way of doing that. Or sometimes you can there might be things downstream ways of reducing the signal itself. So that's my little analogy with desensitization. All right, I think, so metformin, I will walk you through this part. So how does metformin work? Well, it took a while for them to kind of figure out, but the thing is that you have things like the glucose transport. Yep, we know that. All right. So we have this activation and this inhibition right here. But one way that metformin works is to activate AMK kinase right here. So AMP kinase inactivates or inhibits mTORC1. So by inhibiting mTORC1, this prevents it from activating this negative feedback right here. So this prevents insulin receptor signaling from activating, or it will still activate mTORC1 this way, but AMK will prevent that and prevent this negative feedback loop that normally would happen with mTORC1. So, but the thing is that this part right here, this is what I want to show. So even though like mTORC1 is inactivated here, this negative feedback is inactivated, you still have this signaling through AKT, which is important for this glucose transport right there. So that's why even though you're inactivating mTORC1, you still have, at least in terms of like glucose transport, insulin receptor is still being activated. Yep, so that's one way. So little animation. So here I desensitize insulin receptors. And then we add metformin right here. Metformin comes in. And then thanks to AMPK is going to restore and is going to remove that negative feedback. And then now we have these insulin receptors active again. Glucose transports can appear again. And we can get glucose transport. Oh, actually we got through everything. Wow. Okay. So it's time for Kahoot. Oh, I like that. And I like nuclear receptors too. Oh, zebras. Wait, is that like a carol? Oh, okay, yeah. Hormonal Lisa. Wow, this is crazy. I'm glad we turned off like there's a lot of emojis today. Okay. Oh, is that an ox or is that like a cattle? That's so cool. I don't want to remove. So cool. Oh no. Oh, total. Yeah, everyone's making like those AI Ghibli images. Glute 4. Love it. Run.
Run, is that you?
No, it's not me.
Wow. All right. Wow. I'm really competing against myself here, huh? All right, let's start. Oh, why does it say 24, 25? Okay. Oh, yeah. So step 1. So a 36-year-old female G2P1 is undergoing a cesarean section due to breech presentation, so feet first, of the fetus. The patient suddenly goes into hypotension following delivery of the baby. Cardiac monitor show BP of 80/35. So the anesthetist immediately goes to the crash cart and injects ephedrine. Which G protein subunits is catalyzing the effects of the drugs? So the active one that exchanges DP for GTP is G alpha. So it's kind of like Beyonce or Harry Styles. They kind of like become active, go solo, ditch the rest of their group members. So that's what G alpha is. Ooh, okay, more. So a patient diagnosed with type 2 diabetes last year has been unable to establish glycemic control with diet and exercise. You consider starting them on oral biguanide such as metformin. Hey, sounds familiar. How does metformin exert its anti-diabetic effects on insulin receptors? Yeah. Yeah, so it affects that insulin receptor AKT and mTOR pathway. But yeah, it affects it by decreasing mTOR activity. So yeah. So insulin receptor AKT mTOR. So mTOR shuts itself off. So it shuts insulin receptor off, but this AMK inhibits mTOR. Thereby, so this is, you see this all the time in molecular biology, inhibiting the inhibitor, and the overall effect is activation. So it's kind of like the saying, an enemy of an enemy is a friend. That's kind of like inhibiting the inhibitor. Oh, what? Quick.
Oh, the last one, Randy.
Alright, mitochondria. Great. Let's see. Oh, so things that, well, let me just cover this. So we didn't really get to cover so with the typical nuclear receptors for signaling. Okay, real quick. So I like to think of nuclear receptor signaling. So the classic ones are type 1, type 2, and 3. So androgen receptors, estrogen receptors, progesterone, so the sex steroids, they are type 1. And glucocorticoid and mineralocorticoid receptors, So such as one for cortisol and aldosterone, they are type 1. They are the classic ones. And my mnemonic for that is pair up and move in. Type 2 and 3, so these are like the thyroid hormone receptors, and that they're already in the nucleus, but the hormone has to go to them. So I like to think of it kind of like relationships. What do you do when you start a relationship or it gets serious? What do you do? Pair up, move in together into the same house. But what that relationship goes for a long time. Step 2 and 3 is like, do I want to go out of the house? No, I'm just going to get home delivery. So that's the hormone going inside right there. And why did I ask that? Okay, so that is like, here we're showing the type 1. So pair up, move in, activate genes. And let me skip forward to the thyroid hormones. So why did I ask about the powerhouse of the cell? So yeah, you probably saw all those memes, like someone comes up with a meme like every time. But yeah, so many mitochondria is the powerhouse of the cell. But yeah. So there's many memes, but why are they the powerhouse of the cell? Well, back to this equation from like maybe even AP Bio. So you have this, and if you had a really good professor or teacher, so not just ATP, but also heat, because ATP and heat are energy, and in fact, only about less than half of the glucose that undergoes cellular separation is actually converted to ATP. So the rest of the energy is lost as heat. So I think about this like, is every single molecule of gas, assuming you still have a gas car, converted to mechanical energy? No, some of it, this is why engines get hot, right? So some of it is lost as heat. So we do have that receptor in the cell, in the mitochondria, but the thing is that you also have receptors for thyroid hormones in mitochondria. So these thyroid hormone receptors in the mitochondria activate the mitochondria, and by increasing the mitochondrial activity, it's like keeping your engine running or running your engine even faster. It's going to generate more and more heat. So do you need to memorize all this? No, but just letting you know, there is additional receptor in the mitochondria, and that's also why thyroid hormone is super powerful, because maybe other than like your red blood cells and certain other cells that don't have mitochondria, most of your cells have mitochondria. Anything that's able to produce this energy. So if you have a whole bunch of thyroid hormone, you put mitochondria into overdrive. That's why you have heat intolerance. Mitochondria generate heat. So this is why this kind of explains that. Weight loss, increased appetite. What takes nutrients and converts them to energy? Mitochondria. Put them into overdrive, they're going to consume more nutrients. Hyperactivity, now you can bring all these nutrients to energy, so you have like this more energy, anxiety, diarrhea, if you're kind of increasing the motility of the GI tract. And if you're increasing your heat, you're going to get hot and sweaty, right? So that's sweaty, and also you have those adrenergic receptors. And the mitochondria are behind all this. Do they have mitochondria emoji? I think I have to put this in here. But this explains a lot of it. So so this is the way I kind of memorize, like instead of just memorizing this list, just by knowing that the mitochondria are activated by thyroid hormone, this kind of knocks off a lot of the symptoms of hyperthyroidism. Hypothyroidism, pretty much just almost the polar opposite, right? Okay, sorry for going over time, but I just wanted to give you a little tips and tricks. And I'll post the Kahoot later. All right, thank you. Let's see the standings. Ooh, tropical island. Oh. All right, thank you, everyone.